Welcome to this video on deproteinization of biological samples. We will cover the essential methods for preparing samples for clinical biochemistry and analytical assays. We begin with the fundamental concepts, focusing on the definition of deproteinization and the rationale behind removing proteins from biological samples. Deproteinization is the process of removing high molecular weight proteins while retaining small molecule analytes. The core mechanism involves protein denaturation and precipitation, shifting the protein to an insoluble state for easy separation. Protein removal is essential to prevent turbidity and protect instruments like HPLC columns from clogging. It also stabilizes analytes and releases small molecules bound to carrier proteins. A removal efficiency of 99% is often required. Next, we explore chemical precipitation methods, which utilize acids, organic solvents, and heavy metals to remove proteins. Strong acids like trichloroacetic acid, or TCA, and perchloric acid, or PCA, lower the pH below the isoelectric point, causing massive protein denaturation. TCA is highly effective, while PCA is preferred for preserving small metabolites. Organic solvents are also effective. Acetonitrile is widely used for LC megasiemens per megasiemens. Methanol is excellent for removing lipids, and acetone forces protein aggregation but requires chilling. Metal ions and salt precipitation methods are used for specific applications. The Somogyi method removes interfering sugars, tungstic acid is standard for specific blood tests, and salting out provides reversible precipitation, keeping proteins active. Our focus shifts now to physical and mechanical removal techniques, which rely on size-based and thermal separation. Ultrafiltration uses semi-permeable membranes to physically block proteins based on molecular weight. This method is advantageous because it uses no chemicals, maintains a neutral pH, and avoids side reactions with the analyte. Thermal precipitation uses heat to denature globular proteins, and isoelectric precipitation adjusts the sample pH to the protein's pI for maximum aggregation. Finally, all methods require centrifugation to pellet the precipitate. In summary, ACN/methanol is best for mass spectrometry and drug studies. PCA is ideal for energy metabolites, and ultrafiltration is the cleanest though slowest method. These are the image sources used in this video. Thank you for watching.